Hello, my name is Professor Thomas, and today I'm going to be talking to you about mutations. No, not those kinds of mutants. So you may be thinking, what is a mutation? Well, a mutation is the changing of the structure of a gene, resulting in the variant form which may be transmitted to other subsequent generations. But we'll get to that soon. Right now, I'm going to take you through a trip back through time to show you how mutations were first discovered. Let's take a look at this guy, named Gregor Mendel, a scientist and botanist. In the year 1854, Mendel studied plants to find out their heritage or where they came from. At the time, people believed that the features that a life form may have is due to its parents. People also thought that over time a hybrid, the offspring of two totally different species, would revert back to its original formation. That led to the belief that hybrids can't produce future offspring. But Mendel was out to prove them wrong. He used peas to do experiments on. So, what did he actually do? He cross-fertilized them, of course. He studied for a solid 8 years until one day he found something rather odd. He then created two conclusions. He made the law of segregation, which established that there are dominant and recessive traits passed on randomly from parents to offspring, and provided an alternative to blending inheritance, the dominant theory at the time. 2. He then made the law of independent assortment, which established the traits that were passed on independently of other traits from parents to offspring. So now that you know all about how mutations were first discovered, you're probably wondering about how they can occur and what different types there are. Lucky Professor Thomas is here to help, so I'm going to inform you on that too. As I briefly touched on before, mutations occur when a genetic modification is passed on from one or two parents, or hereditary mutations are inherited from a parent and are present throughout a person's life in virtually every cell in their body. Now let's move on to the different types of mutations. We have substitution, the change of a codon that embraces another amino acids and causes a minor change in the production of protein. Insertion, extra pairs of DNA are inserted. Deletion, vice versa, and the frame shift. Since protein coding DNA is divided into codons 3 bases long, insertions and deletions can alter a gene so that its message is no longer correctly passed. These changes are called frame shifts. Now moving on to our final subtopic for today, the future of mutations. Is there a cure? Unfortunately, some are irreparable, but some can be. Here's an example. For a group of genetic conditions called inborn errors of metabolism, which result from genetic changes that disrupt the production of specific enzymes, treatments sometimes include dietary changes or replacement of the particular enzyme that is missing. And then it leads on to say that it can in fact be treated. And as the future for mutations? I don't know. No one really knows. Scientists are working on mutations day in and day out, so hopefully in the near future there will be a major breakthrough. What are my thoughts on mutations? I say they are really fascinating and some are rather cool. You look at the way mutants and mutations are represented in the media, whether it be fact or fictional, is pretty great. What have we learned today, students? What a mutation is, how they first came about, 2 different laws of mutations, types of mutations, if there is a cure, and what the unpredictable future has in store for us. Thank you for watching. See ya!